Hello everyone. So today we will discuss about G protein coupled receptors. G protein coupled receptors. G protein coupled receptors are polar signaling molecule receptors, and these are plasma membrane receptors, as you can see here. And G stands for what? G stands for guanine nucleotide binding proteins. So these are generally transmembrane proteins. They have they have a typical structure. So G protein coupled receptor are large family of proteins that has transmembrane helicase. You can see here. This is transmembrane helicase. So this is transmembrane helicase, and this whole 1, 2, 3, 4, 5, 6, 7. So these are transmembrane helicase, and this is ligand binding region. So transmembrane helicase is a bundle of 7 transmembrane helicase that forms the core of the receptors. This is the core of the receptors, and there are 3 extracellular loops and there are 3 intracellular loops. You can see here E1, E2, E3 are extracellular loops, and I1, I2, I3 are intracellular loops that connects those transmembrane helicases are connected by these 3 extracellular, 3 intracellular loops. And there is in ligand binding region there is N terminus where it is an amino terminus. It is extracellular amino terminus, and there you can see the C terminal where intracellular carboxyl groups are attached, and there G alpha G proteins are linked to the C terminus side of the receptor, and these G proteins are G alpha, beta, and gamma. This G alpha proteins and beta and gamma, those proteins are attached to the C terminal region of these receptors. So signals of these G protein receptors, like it helps in vision, olfaction, taste, glucagon receptors. Those are G protein receptors, and luteinizing hormone, epinephrine, acetylcholine, serotonin. Those are the examples or signaling examples that bears G protein coupled receptors. So here we can see that G protein coupled receptors, G alpha, beta, and gamma, those are attached with the C terminal end of the receptor, and this C terminal end, and there is a GTP bind with the G alpha, and this G alpha GTP binding makes this G protein in active state. And this due to active there is a dissociation of G alpha GTP. There is several functions of different functions of those G alpha, beta, and gamma. So alpha, beta, and gamma. So that's why there is dissociation of G alpha GTP. Here you can see that G alpha GTP dissociates from beta and gamma, and those have separate works, like here G alpha activation of target enzymes. They can activate those target enzymes, that is adenylyl cyclase, phosphodiesterase, phospholipase, C beta. We can see those enzymes are activated, and these are discussed in further mechanisms. And ion channels activation, potassium ion channels, those are discussed in further mechanisms. And there you can see G alpha subunit. So G alpha subunit are mainly 4 types. Those G alpha subunits like G alpha sub S, that is stimulatory, G alpha I, that is inhibitory, and G alpha Q, that is phospholipase C beta, and there is G alpha T, that is transductions. So these alpha types are acts as various functionings. They have various functionings, like some are like that stimulatory, activates the adenylyl cyclase, whereas we can see that inhibitory G alpha I, that is inhibitory, inhibits the function of adenylyl cyclase, and therefore CAMP synthesis not occurs, whereas we also can see that in G alpha Q there is activation of phospholipase C beta enzyme. So that also another types. And another was transduction G alpha to T, that helps in phosphodiesterase activation, which leads to the activation of GAMP, formation of GAMP from CGMP to GMP or CAMP to AMP. So that is the subtypes of G alpha subunits. So those subunits are discussed in the next part.